And nice to meet you at this Friday evening. I can act past, network, and unit. We met here every Friday. So this is hello from Beijing. I think good morning, good evening, good noon. Yeah. So everywhere this work was almost more than 1 year. So we met a lot of scientists on this stage. So today it's my great honor, to host this session. So today the speakers is my great friends, and the panelists and the challengers are all my friends. So yeah, today our talk will be very hot topic. It's from Professor Yanyi Huang. It's going to deliver talk for Friday evening. Strengthening for the network and unity. And Yanyi was a superstar. So this is hello from Yanyi looks like a high school student. Yeah. So everywhere this work was professor for more than 1 year. So we made a lot of the best professor. So today is my great honor really done a lot of work. And so today the speakers is my great friend. And we heard about the panelists and the challengers are all my friends. So today our talk will be very hot topic. We're going to hear from Professor Yanyi Huang. How can help you deliver talk on Friday evening molecular strengthening for the network and unity. And Yanyi was a superstar. So this is hello from Yanyi looks like a high school student. Yeah. So everywhere this work was professor for more than 1 year. So we made a lot of the best professor. So today is my great honor really done a lot of work. And so today the speakers is my great friend. And we heard about the panelists.
To present some of our recent work at IKX. I know the audience must be extremely diversified, so I will try to make this talk as interesting as possible for most of you. I'm a professor of chemistry working at the Peking University. I'm also PI working at a few interdisciplinary research institutes that allow me to explore to many other fields and interact with many scientists with very different backgrounds. Today I will focus on some recent progress of my group related to nucleic acid sequencing and the applications in single-cell studies and the fighting with coronavirus. This ongoing pandemic has inevitably changed our lives, and I missed old days a lot. I guess you guys have the same feeling. Say this background from this slide. The picture was taken in my university. The campus is beautiful. It was taken just before we met this new virus. This peaceful and joyful time now became one of my great memories. My group is working on a few fields. If I have to summarize all the works into several categories, I think that these four may work. Microfluidics, bioimaging, small amount of nucleic acid sequencing, and the error elimination through a method we developed called the ECC sequencing. Today, I will only touch a small portion of these works, and I'm sure these I'm going to talk are more than enough to showcase the style of research done by my unique students and postdocs. As many other groups working on microfluidics, my group has been developing many kinds of chip-based devices, which means you like to put some functions of liquid control into a miniaturized chip-like device. It has another popular and somewhat fancy name called a lab on a chip, meaning you can put laboratory work pipeline or at least a few process steps onto a chip. I've been keen on designing and fabricating all kinds of such lab-on-a-chip devices. For example, we convert the microfluid devices into a dispenser long ago. Right? So now you can actually transfer by cutting the channel open and perform nanoliter accuracy liquid transfer. Now you can do this liquid transfer between devices. We also figure out a way to perform quantitative chemical analysis through inaccurate actuation, such as the finger squeeze, like this work here. and we have made the leak transfer back and forth through multi-layer design. We also couple light into the liquid channel to make optical detection integrated seamlessly with the fluid flow, so on and so forth. We also tested how to culture live stuff from cells to small animals on chip. For example, we can culture cells or various types of cells together on a chip and observe their motion for a long time, say days to a couple of weeks, and assess their mobility with high precision and accuracy. We also culture C. elegans, and this can be done for weeks, observing their whole lifespan and see how genetic alterations or environmental conditions affected their aging process. We even culture the zebrafish. We culture zebrafish embryos for days and see how the drugs can cause developmental defects. Recently, most of my students are not working on such type of lab-on-chip device I mentioned before. But in my group, there's always one or probably two persons keep these conventional skills. Last year, through collaboration with Professor Chen Zhang, through Han Gui in my group, has published an interesting paper describing how to monitor the circadian rhythms at the single cell level using microfluid devices for cell culture. This is a simple microfluid device in terms of fabrication. What you see here is a set of 4 thin chambers for culture mouse adult fibroblast cells. Actually, a few different cell lines in which some genes are knocked out to test the proper functions of a circadian clock in using these cells. The device was made of PDMS, my favorite material of making microfluid devices, circuits. The chip is optically transparent, so we can put this device inside a miniature culture incubator that can sit on a microscope's translational stage and take an optical imaging through a microscope with the engineered cell lines. The bioluminescence signal can directly reflect the phase of a circadian clock. Hence, we can analyze the optical image and retrieve this circadian rhythms of each cell. We can take advantage of the microfluidics precisely introducing VIP. It's a vasoactive intestinal peptide, which can reset the clock of these cells. Within 6 nanomolar VIP triggering every 24 hours, the second clock can be maintained for much more cycles. I will not talk about the biological findings of this work, but just mention that by checking each cell's phase of circadian clock, we clearly notice that through long-time culture, the cell will gradually lose the synchronization of their individual clock. However, if you add 60 nanomolar VIP every 24 hours, you will see the reset. You will see the reset of clock for every single cell. Quantitatively, this phenomenon can also be assessed through a parameter called SI, the synchronization index. Theoretical addition of the VIP does indeed boost the synchronization index of the cell community. Now, let me get back to focus on today's topic. I'm a chemist, right? So when I look at any life system, the first impression that I compared to chemistry or physics is the enormous complexity. For instance, every single cell are different from their neighboring cells, showing great heterogeneity. Also, with the development or aging, one can also observe temporal heterogeneity caused by genetic changes over time or cell division. Such complexity also happened on many diseases, such as the most common example we like to use here, cancer. Understanding such complexity requires high sensitive and quantitative measurements of the genetic materials at the single cell or similar level. Even within each single cell, the complexity is still quite amazing. Genetic materials, including DNA and RNA, contain a lot of information on cells' status and identity. I will start from DNA. Each single human cell contains about 6 kilograms of the DNA, which is just 2 copies of genomes. One copy inherited from the father and the other copy from mother. Now, in biology and medicine field, people are interested in interested to know if we can find the genomic difference between each cell through the high throughput sequencing technology that becomes more and more popular and affordable these days. To give you some examples how a technologist like me working on single cell sequencing, I'll start with a strategy we call the divide and conquer, which was an old war strategy that has been highly recommended by a famous ancient Chinese military treatise called The Art of War, written about 2,500 years ago. Almost 10 years ago, 3 of my students, 1 senior one and 2 junior ones, conducted a very straightforward experiment, trying to push the volume limit of single copy PCR using a unique microfluidic approach. The quantification idea was digital PCR, which was not very novel, I must admit. However, we still challenged ourselves by pushing the technical limits. We manufacture very simple microfluidic devices here. Showing here that it has many microvials in this 2 by 2 millimeter area. You can put about 80,000 microvials. We also set up a simple thermocycler here to couple with this PCR machine devices. Digital PCR is a quantitative measurement that can assess the target sequences and has the ability to absolutely count the copies without using any reference materials. So it became more and more popular these days. One of the technical limits we pushed very hard was the reaction volume of each compartment. Our well is only around 36 femtoliter, which is much smaller than the most other digital PCR approaches using the picoliter or nanoliter size compartments. Under a microscope, we can image the well, and the bright ones indicate the successful performed PCR reactions, while the faint wells mean the PCR reaction, the reactions do not happen there. Hence, we can count how many wells are positive. And each positive count means that there is at least 1 copy of target DNA fragment sitting inside a well. The chance of more than 1 copies of DNA sitting in the same well, it's there, but it's low if the total counts are significantly less than the number of the wells. Right. For example, below 30%, then follow the Poisson distribution. You can do the statistics. Then most wells, the positive ones, will contain 1 copy. So you can do absolute counting for the digital absolute counting for DNAs. Hence, we can just simply identify the positive from negative wells and then obtain a number of targeted DNA molecules by this very simple image processing, and deduce the concentration given the volume of samples. So you can do very similar diagnosis as any other PCR methods. When we looked at paper, that's the most important part. We had a chance to write whatever we want, basically, right? So we put a paragraph of somewhat less related to the other part of the manuscript, describing a thought that we had at that time. So this such divide and conquer approach, we probably can make application bias reduced. So we did not perform any experiments at the time, just put what we had in our minds into a paper. It's like writing a diary, and hopefully one day later we would come back to rethink about this idea, right? Not long later, actually, we start to re-evaluate this idea when we try to sequence a single cell's whole genome. As I have mentioned a moment ago, each single cell has only 2 copies of the genome, and it is extremely challenging to sequencing them directly. Since DNA can be faithfully copied, we typically amplify the single cell's genome to a large amount first, and then we perform the sequencing. Most single cell whole genome sequencing results are used for obtaining 2 types of the genetic variations, copy number variations, which means the abnormal copy numbers of certain location go through the genome, right? And the single nucleotide variation, which is the most common genetic mutations. To make the whole genome amplification really feasible, we need to consider 3 major aspects. First, evenness. We want every position of the genome amplified with the same efficiency then the information of copy number variation can be correctly retained in the amplified products. And second, completeness. Since each single cell can be amplified only once and it's gone, and each cell is different from the others, so you have to amplify single cells whole genome as much as possible. So we did done completely. And third, accuracy. This is easy to understand since we are sequencing the application product of the original genome. We want every base of the genome faithfully copied into the replicated ones. Otherwise, we will read the wrong information. There are many existing methods for single-cell whole genome application. For example, one popular method called the DLP-PCR that has greater evenness but very low completeness and low accuracy. In 2012, Professor Sanish's group invented the MALBAC, a new method include quasi-linear application steps to significantly facilitate the completeness with excellent evenness, but still suffer from low accuracy. On the other hand, MDA, or multiple displacement application, another popular method, can amplify single-cell whole genome with isothermal reaction, can offer greater accuracy due to the usage of high-fidelity 529 primers. But the evenness of MDA is notoriously bad. Many researchers in the field, in my field, like doing microfluidics, try to implement the microfluidic into MDA reaction and use small volume to suppress the amplification to reduce the bias. But these attempts usually cause the decline of completeness, and hence not an ideal approach. You have to pick one over the other. Too bad. Yu Si, remember she was the junior graduate student in the previous project, now became a more experienced senior one. She did some conceptual thinking. When we perform an MDA reaction in a conventional way, say in an epitope tube, it happens like this. If an original copy number of each fragment is 1, say here 1, 1, 1, 1, right? In the beginning of the reaction, some fragments will initiate the reaction quicker than others, probably due to the sequence or due to the stochastic triggers. And these fast reacting fragments will finally be amplified much more times than the slower reacting ones, causing significant bias of the MDA reaction. If we separated reactants into many segregated small reactors, for example, small droplets, the MDA reaction is still isothermal and easy to operate. However, in this way, the faster reacting fragments will amplify early, but eventually reach a situation where the slow reacting fragments will eventually catch up. So after the same amount of time and reaction time, all the fragments reach a similar gain. Hence, we increase the evenness of MDA without losing the completeness or accuracy of it. We first tested the performance of emulsion MDA idea using single primary fuel-back cells. These cells are diploid cells. That means only 2 copies of DNA. And the donor is male. So from the bulk sample, you can easily see the copy number of the chromosome X and Y here, right? So about half the copy number of the autosomes. These 2 copy numbers, 1 and 2, can be easily separated, as you can see from the content density histogram. Conventional MDA are extremely large random bias. Copy number identification of any specific location of this genome can vary across over 2 orders of magnitude. And this copy number of a sex chromosome and autosome are not able to be differentiated. MALBAC does provide an evener application, but indicated here, not surprisingly, emulsion MDA provided the evenest sequencing result, which is closest to bulk. So I will skip the details of the numbers. So in summary, through a simple divide and conquer strategy, single-cell whole genome application can reach high fidelity, high coverage, and high uniformity simultaneously. Emulsion itself is generated by a simple microfluidic chip. The emulsion is very stable. You can clearly see the phase separation between oil and aqueous droplets in the tube, right? This is the experimental setup. I took this image with my cell phone in the lab. It can be easily adapted by, I would say, by any biological labs. It is actually the simplest ever microfluidic devices that my lab has made in the past, say, 15 years. However, most biologists still think such simple microfluidic setup is not ready for them to use. So too bad. So they want something extremely simple and easy to use without so many tubings, connectors, pumps. I actually think what I think is very valuable. The good technology needs to have the potential to be easily transferred from lab to lab, from lab to bedside, and even to the end customers, right? So the other junior student that got involved in this in the paper I just mentioned, the 2012 paper, was Zhi Tian. He also became a senior student a few years later, right? So in his spare time, he actually developed two methods trying to solve this problem. First method is called SIMPLE. This is a fun project. His first pilot study was done actually by taping pipette with an electronic toothbrush and disperse aqueous solution into oil while stirring the capillary by turn on the toothbrush. Now they deliver the aqueous liquid into the oil. Then amazingly, with almost identical physics that used by microfluidic water in oil droplet generation scheme we just showed using the microchip, we present a much simpler idea without using chip. One nice characteristic of our new approach is that since the oil we used is lighter than water, so the droplet would slowly sinking down to the bottom of the container and will not be further smashed by the moving capillary. While we thought that this simple method is a nice invention that the biologists will enjoy to try, we still felt it was not elegant enough. Later, Zhi Tian developed another method that we call the mica, which I believe is far more elegant and simple. The key component is a small glass plate in which there are a few small holes. In this picture, right? In this picture, you see 7 small lines. They are actually small holes embedded in the plate. Each hole or each channel is 6 micron in diameter, and the length is the thickness of the glass plate, typically 500 micron. This tiny glass plate was made by ancient technology. The glass rod made of different materials. Some can be etched away, are piled with others, and then refused by heating those rods together and drawn into small glass rods, then sliced into identical pieces. The holes are then formed by ester etching, and surfaces were coated through CVD to be hydrophobic. Hence, the aqueous liquid placed on the glass plate would not dripping through the microplate due to surface tension. When you put the assemble into the centrifuge and spin in a very fast speed, at least 5,000 G, then the aqueous solution will flow through the channels and then jet into the air, become a small droplet, and then submerge into the oil. When there are proper surfactant in the oil, the droplet will form water and oil emulsions, very stable. As you can imagine, different spinning speed generates different size of the droplets. It is so convenient that literally everyone in the group can just use a tabletop centrifuge and hit one button and generate uniform droplets. Meng Chen, another student, then was just an undergraduate, designed a new swim bucket. Then each one can take 4 or even 8 small tubes with the mica plate inserts. This way, we can easily increase the throughput to 24 or 48 generators in a single experimental run. each tube can be used for amplifying one single cell's whole genome. And then we can test the performance using sequencing technology. So sequencing results clearly show that similarity of each single cell when the cell line is used as input. MICA can generate as good, if not better, results than using chip-based microfluidics. However, number of droplets is critical. In this paper we published 2 years ago, we have optimized its experimental parameters. That's all those parameter space. So by varying these parameters, we are generating the highly even applications and optimize the whole protocol. In summary, my group has been working on combining microfluidics ideas and concept with a single cell whole genome application and sequencing for years, and gradually transferred from lab on a chip format, as I show here, to lab on a chip, and then we move into lab off the chip idea. We use the powerful centrifuge-based emulsion generator to improve application evenness. We still seek for better, the simpler way to amplify single cell whole genome. We're still working on this in this field. But I want to switch a little bit to the side product while we explore the single cell whole genome sequence. That is, we can seamlessly use these droplets for digital PCR. As we have summarized in the picture, although we have made the PDMS-based micro the smallest micro digital PCR and push the size limit to the very extreme, we found that the droplet we generate actually with a size of a few tens of microns are probably the best reactors for digital PCR. We recently published a paper describing the optimization process of finding the suitable oil and surfactants for stabilization of droplets generated through this MICA idea. We aim to provide a good recipe for generating both thermally and mechanically stable water in oil emulsion for PCR reactions. If any of you are interested in trying such emulsion, you are welcome to read this paper written by Fang Li. All details are there, the recipe is open. The droplets are very stable with identical size, making them perfect for digital counting, especially like PCR. And using proper probes, the successful application is any individual droplet will generate a positive fluorescence signals for indication of the existence of the target molecule. However, although we have cleverly invented the MICA to overcome the difficulty of using unfamiliar microfluid setups, so now the generation can be done using a centrifuge, and the application itself can be done using conventional PCR machine. You don't need an actual machine or some specialized machine. But the counting process is still not easy to be done without some technical challenges. In our 2017 paper that first described the MICA idea, we actually hacked a second-hand flow cytometer. I bought a cheaper one, the second-hand one, to let all emulsion drop the flow through a big capillary after application, and then count the single intensity of each droplet. This is, of course, working well, but it's not elegant enough. I don't like this three-step approach. More annoyingly, this counting strategy required the tube lid to be open and then transfer emulsion into another machine. We hence faced two major risks. First, the opening step may cause cross-contamination since the targeted molecules have been amplified by thousands to millions fold. Second, the transfer may cause sample loss, and for very low copy detection, this may cause big uncertainty in the result. We have a solution to perfectly solve all the questions and eliminate those risks. And I like to quote Dao De Jing, the ancient Chinese philosophical classic for Taoism. The great form is without shape. Recently, my student Pei and mentioned, as well as a former student Zitian, collaborating with Professor Yanyi Huang from Tsinghua University and Professor Fei Peng. Peng is an optical imaging professor from Huazhong University of Science and Technology. We published a paper describing the details of the newest toy we built in the lab that we can do digital PCR detection with amazing performance. We call this method 3D clear digital PCR. And the major technical advantages are lossless during both steps of droplet formation and droplet counting. The key idea of this invention is to match refractive indices of aqueous solution and the receiving oil. Using a centrifuge-based microspectrometry to generate droplets emulsion is easy. While what we usually describe as emulsion is an emulsion, we typically would expect a milky liquid form due to the light reflecting. You can see this multi-step due to light reflecting in those interface between oil and aqueous phase. But tuning the index of either oil or aqueous phase to match, if we can make them close, right, then the milky liquid will change to apically clear, even the emulsion still exists. So the goal is not just making the emulsion optically clear, but such optically clear sample can be well imaged, three-dimensional, through a simple light sheet imaging scheme. To check the positive versus negative droplet after a few tens rounds of PCR application, typical 30 to 40 rounds, we apply this conventional use the fluorescence reporter, such as the Techno Pros. Unlike the normal use the series detector that count one by one droplet or imaging a single plane of the spread droplet, now we can just put this tube in situ detection. We focus the excitation light beam into a sheet with a thickness of about 20 micron. It can scan through the sample, and each section is imaged by a camera. So the sample itself can then exact PCR tube that can use for generation of droplet, and then the same tube can put there for imaging. Therefore, there's no sample transferring is needed. This not only eliminates the loss of the molecules, but also prevent the possible cross-contamination, which will cause annoying false positives for such a sensitive method. Of course, we can scan the PCR tube by different excitation wavelengths and emission wavelengths and obtain multiple channel images, or all three-dimensional, very quickly. Right? But we built a few lab prototypes for having fun with this light sheet imaging of the whole tube emulsion. This is one of the prototypes we built, and the size is about a shoebox size, and almost all the components can be directly ordered from Thor Labs. Very easy. We ordered the parts online, and we actually received them in a few days, and then we spent a few hours, literally just a few hours, to build the whole system. This is a light sheet of this laser. It's a 473 nanometer laser, and that's the PCR tube. You see the light sheet shining through the PCR tube. Right. When the laser excited through the tube, in which emulsion has been generated, sitting quietly in the tube, you do not see much scattering. They just go through it. Right? While you observe through you observe through this emission filter. Now you can see you can observe this fluorescence generated from the positive droplet. You see these blinking droplets in excitation plane, and importantly, emulsion is still very robust. They don't merge together. Even after adding many additives to tune the red indices, you see these droplets through this imaging plane. And droplets did not coalescence to each other, nor delay move during the scanning, which takes only a few seconds to cover the whole tube. So two channels are recorded separately, and positive and the negative droplets are clearly differentiated by their fluorescence intensities. If you want, such a three-dimensional image can also be merged together, right? As you see in the right side. So each scanning takes only about 4 seconds. Although it is quite easy to count the positive by manual screening and image sectioning with naked eyes, it's not that sophisticated to develop a robust algorithm to count the positive droplets. The raw images acquired by CMOS camera can be background corrected, then denoised to provide clean pictures, and then we count the 3D reconstructed data for positive clusters. This 3D image reconstruction and the positive droplets counting method has great potential to accurately assess the nucleic acid copies from 0 to over 100,000, which is about like 6 log dynamic range. This is important. Such dynamic range is as good as conventional quantitative real-time PCR assays, and better than currently available commercially available digital PCR machines. In addition, thanks to digital counting, we can reach a much higher accuracy by having much starting uncertainty of measurement compared with other existing qPCR or digital PCR methods. After all, this is a fun project. We all have so much fun by inventing something that's practically useful, and the simplicity of this method is what we really enjoy. And hopefully one day digital PCR can be as easy and as cheap as conventional qPCR assays through these methods. A few months earlier this year, we had another paper describing another approach we developed later to perform in situ imaging analysis without opening the lid after application. After droplet generating using mica, mention, insert a rod inside the tube, and then flip the tube to let the droplet sinking down to form a thin layer of packed droplets surrounding the rod, in between the rod and the inner surface of the tube. Because there are only about 3 to 4 layers of droplets in this layer, the imaging can go through the droplets very easily with no need of index matching. So save another step. These are a few pictures of the real devices and the imaging setup. Once the tube with droplets flipped upside down, within about like 20 minutes, the droplets will all sit in the bottom and beautifully packed. And a line shape of excitation it's used with rotation. We can collect the image of all droplets. The image analysis is straightforward. Stitching many small slices of image into a bigger one, and clean the background and denoise, and then count the positive spots, as you can imagine. So we have demonstrated that in this new method also have great dynamic range, excellent linearity, which make it another good choice for digital quantification of nucleic acid. So in this paper specifically, we used LAMP as a thermal application method instead of PCR to demonstrate this whole idea. We also work on RNA. A cell contains about same mass of RNA compared to nuclear DNA. We actually invest a large amount of time and efforts on RNA sequencing. We mainly focus on the development of new methods to construct the high quality libraries from low input of RNAs for sequencing. So today I will show only one topic that I like to quote from Confucius: sharp in action. Right? You will see why I mean this. A few years ago, the same student, the one who initiated the single cell copy number alteration study doing the emulsion MDA, right? She also spent quite some time trying to figure out a way to simplify the library prep of RNA sequencing. Recently, dealing a graduate student in my lab, together with other students in Jianbing and Sunny's labs, published a paper presenting this super simple method we call the sherry. It's coming from the name of a wine. We are trying to find a new way to sequencing RNA molecules from the small amount of starting material. The existing methods, for example, this one widely used by many researchers, are time-consuming and labor-intensive. The idea is simple yet wild in the beginning. We are trying to do. In the past decade, people are getting more and more interested and getting very popular to use an enzyme called the TN5 transposase, an engineered molecular machine, to cut and then tag double-strand DNA on each fragment to make the sequencing sample preparation much simple, easier. Right? We then ask ourselves the question: can we borrow the same idea and use TN5 transposase to directly cut RNA DNA hybrid duplex? After we do the reverse transcription and without second strand cDNA synthesis, which is not what we really enjoy to do, right? Because if so, then the RNA sequencing will be revolutionized. People have long known that TN5 transposase belongs to RNase-H superfamily, and the activity sites of TN5 and other RNase-H are very similar and conserved. We then experimentally demonstrate that TN5 transposase can indeed cut and tag the RNA DNA hybrid duplex. My friend, Professor Dong Wang at the UCSD helped us checking the structure of TN5 transposase and encouraged us by saying, hey, see, this enzyme can properly process A form heteroduplex with similar configuration to process the B form DNA duplex. Right? When we experimentally confirmed that cut and tag functions both worked for RNA DNA heteroduplex, then the rest became easy. The whole process from RNA to a sequencing library can be finished within about 4 hours with hands-on time about only half an hour. So students will love this protocol. We also proved that Sherry protocol has a wide dynamic range of input RNA. It can work across 6 orders of magnitude from bulk to single cells. I will not go through all these details since it has been published, and many people probably wouldn't really get the sense just waiting a few minutes. So you are welcome to read the paper if you're interested. You're welcome to test by yourself. It's so easy. We also briefly test the single cell inputs and see a very promising potential. We, and probably many others, will keep working on this direction, seeing if such simple idea can be further improved to single cell RNA-seq. The most desired range, the purpose we originally designed Sherry is not for single cell. It's around 100 picogram RNA input, particularly due to the lack of good existing protocols for such amount of RNA inputs. We believe Sherry is becoming the simplest RNA sequencing simple prep protocol for such we call them mini bulk samples. Well, when this paper was about to publish, we had a completely unexpected test, a real-world test, the COVID-19. Based on Sherry, this protocol, we made some modifications. The true modification is here. We added some randomers as the primary here. Also, a few other details. We developed a new strategy called the Minerva to utilize the simplicity of the direct segmentation of RNA DNA heteroduplex and to work on the clinical samples of COVID-19 patients. The samples can be pharyngeal swabs, sputum, or even stool samples. Within about 4 hours or so, we will have the library ready to send for sequencing. This library here we call the Minerva M is the meta-transcriptomic library containing both pathogen and host signals. With specific probe enrichment steps here we described in the bottom. In a few more hours, we can generate an enriched library. In this case, we enrich the SARS CoV-2 for identification of the mutations and specifically intra-host single nucleotide variations. Our collaborators, Professor Zheng Hui, Professor Chen Chen, they're infectious disease experts working at the Ditang Hospital, which is the major hospital in Beijing to treat local COVID-19 patients. And Jianbing and I were working on the protocol on Angela at Hong Kong University of Science Technology, is taking care of the analysis of meta-transcriptome. This is a very pleasant collaboration between 4 institutions. We used 137 samples from 91 patients to test our Minerva protocol. This is in last year is in March and April, right? And the whole process is much easier than the previous existing method that uses double-strand DNA ligation to construct the library. Using Minerva through metatranscriptome data analysis, we can clearly see the disease severity is strongly associated with the microbial composition. This is interesting, but we'll skip the detail, and the paper has been published, so you're welcome to read the details there. Right? I want to emphasize only one thing: through the viral enrichment, despite the type of samples, you can be stool, be sputum, be pharyngeal swabs, we can obtain complete or nearly complete genome sequences from majority of the samples. Compared with conventional double-strand DNA ligation protocol, Minerva is more efficient and more reliable. I'll give one interesting example on capacity to identify intrahost SNVs, which may give the hint on the virus evolution in your body. From a mutation profile, we can easily find all the mutations happened across the genome. There's some hotspot mutations there, right? Including the linkage between these mutations. So these arrows here, the blue and the red arrow, indicate these linkages between these mutations. The mutation of the this one, like 87, 82 here, and also the 28, 144. These 2 positions are linked together. That means the mutations are either mutated, they mutate here, they have to mutate the other position. They are linked. And these mutations are the one defined by my colleague, Professor Lu Jian. They call it SNL strains. And that are also correlated to disease severity. The other 4 linked mutations in this blue arrow indicates are especially important since last April. They already become the most popular signature mutation in the virus worldwide. We posted the manuscript online on last April, and we have written a very detailed protocol to go with this manuscript. The protocol, if I have to summarize the major advantage, is easy and robust. This is very important. And indeed, very practical, especially during pandemic. No more spare labor or energy available to work with complex protocol when all the medical and the related facilities are busy working with patient treatment and virus containment. This Minerva method has been used to perform the detailed analysis of the first several waves of local outbreaks in Beijing, and clearly show the difference of domestic wave, the first blue here, domestic wave, and also the imported wave, the second wave, with the different genetic signatures. Let me show you a very special applications of this Minerva protocol. In last June, about a year ago this time, Beijing was hit by a resurgence of COVID-19. During that time, the origin of the virus was unclear. So Jianbing and I have worked with a lot of people, majorly the experts in the Institute of Pathogen Biology of Chinese Academy of Medical Sciences, Institute of Genomics in Chinese Academy of Sciences, and also the Beijing CDC team. we quickly sequenced over 100 key samples, both critical patients and contaminated environmental samples. It was very difficult to extract and sequence those RNAs, and we managed to obtain 72 high-quality genomes from these samples. To make a long story short, what we found is that through an integrative study that combines viral genomics, sequencing, and a large-scale population test, as well as the very thorough epidemiological investigations, we can map the viral transmission route with high confidence, and then found that the imported seafood, actually salmon, through international cold chain logistics, is highly possible the origin to transmit the virus through a long distance. Our findings are particularly important for countries where community transmissions are contained or suppressed, like China. The virus could be reintroduced through cold chain transportation of contaminated items and might initiate an outbreak. Even with low probability, such a viral transmission would have caused large-scale outbreaks if not intervened immediately after the first cases. So regional guidelines on COVID-19 prevention and the control should integrate surveillance of the cold chain imported products, foods, seafoods, especially those from epidemic regions for COVID-19. Due to time being, I'd like to stop by showing this famous poem written more than 200 years ago by William Blake. I was deeply moved by the philosophical meaning in it. When we are working in a lab, actually all small findings of many small detailed phenomena. But every small finding is important when you link them together, both temporarily or spatially. You will build a beautiful world with deep understanding. About 100 years ago, a famous artist monk, Hongyi Fashi, has translated this poem into Chinese. A beautiful translation for those of you who understand Chinese. Let me thank all of my current and previous students, postdocs, and collaborators who I have privilege to work with and to make those small findings. I just shared with you. I only have time to share a very small amount of these findings. And also I will not go through all the names because of the time limit. Thank you all for having me here. I sincerely hope soon we can see you in person. You're welcome to visit Peking University. It's a beautiful campus. Thank you.
Okay, great, Yanyi. Wonderful talk. Congratulations. Very nice. Thank you for using your technology to connect the world and the universe, and also you save us, right?
Shall I stop screen sharing? Right?
That's okay. Yeah, you can share the stop that. And now welcome everyone to get on the stage to do our panel discussion. Let me first introduce one by one. Wenchang Hu, Walter Hu, was from Sichuan. Actually, before he moved to Sichuan, he's a professor, a full professor in UT Austin, right? Dallas, right? So Walter, you mute. Yes, welcome, Walter. And our next panelist is Xinyu Liu. Xinyu was a professor in Canada. And now he's in University of Toronto, right? Xinyu was done a lot of work in microfluidics. So I think he will ask more questions today. And our challenger here is Dr. Hao Tian Chen from EPFL. Actually, Hao Tian looks more nervous, So, let's welcome all our panelists on the this panel discussion, because today the topic is a really hot topic. I think most of people was really, curious about this, how to detect all these viruses and, how to, it's still there, it's still changing almost every day. Yeah, so Hao Tian, I think first chance I give to you as extra challengers, you can ask the questions first, please.
Okay, thank you very much. This is Hao Tian Chen from EPFL, Switzerland. I would like to thank first to Alex, Professor Zhang for providing me this opportunity to have discussion with all the wonderful professors here. And thanks Professor Huang for their very impressive talk. I really enjoyed it. Actually, here I have several questions. So for different, I noticed that you develop a lot of tools or methods to like separate the cell, a group of cells into different locations. I'm thinking for different type of cell, because they have different form, size, or feature, is it possible to use the same method to do the same, to achieve the same goal? For example, I mainly work on the neuroscience related bioelectronics for the neuron. Its size is very different from the normal cell, like liver cell or heart cell. It is possible to use the same method to separate them. Okay, that's my first question.
All right. Well, it's actually quite a hard question for me because I never really cultured neurons in my own group. But I do have many collaborators and friends that are working on neurochemistry, neurobiology. So culture neurons is not very easy first. So for neuron culture, typically you require more efforts to optimize the condition. Also, sometimes you actually need to use microfabrication to assist neurons to grow, to direct the axon guide, reach certain positions, and also to build a neuronal network if you want that. And for biological studies, sometimes you actually like to analyze the single neurons. For example, you do single neuron sequencing, or you do single neuron detection, you do patch cloud. Because the size of neurons, they actually vary quite a lot.
Yeah.
And also the axons, they entangle together one to another. Right? So in the tissue, they actually can spread everywhere with a very long axon. So it became very difficult to separate a single neuron without damaging it. Or even you don't damage it, you actually probably alter their status. So when you measure it, it's not what you want to measure sometimes. So from analysis point of view, we sometimes cannot figure out a best way to separate a single neuron. Then we separate nucleus from the neuron, because we have enough evidence for neuron, especially single nuclei sequencing can present most information you want, especially if you are interested in the cell typing. So that's why when you do the large throughput, a high throughput, large scale single cell study, you sometimes do single nuclei study instead of single neuron study, because by doing neuron separation, you will cause a huge bias, because you probably previously select larger ones or you select smaller ones. You select intact ones, you select broken ones. But when you do nuclei study, then you actually avoid this sampling bias by taking all the nuclei and do single nucleus study. So that's one of the detours we used in single neuron study. But you are absolutely right. For those micro scale tools, sometimes we are actually causing a lot of biases by using these tools, because by screening, we actually have preference to select certain types of cells we want or we don't want. then so the cell counting became unreliable at this in these studies.
Okay. Then is that answer your question?
Yeah, thank you very much. And another, a very quick question, because I think the way to separate based on like the oil peptide generator, this type of is very cool. But I mean, I'm not very inside this field, but I heard that some technology like self-assembly is also a very cool way to separate like micro or nanoparticle. Do you think it's all possible to apply like this technology to separate cells by engineer the structure of the substrate, for example, engineer the structure of the PDMS and code with some spatial chemistry or some other material. So after the fluid of the cell flow from this substrate, the structure can capture or absorb the cell by itself. Do you think it's possible to do this?
No. Thank you for it's a great question. Yes, the short answer yes, it's possible. In microfluidics field, there's been at least 2 decades people working on all kinds of the strategies to retain, to separate, to pattern cells or single cells or cluster of cells. So there are probably 3 major, I would say 3 major approaches. If I just came off my mind right now. First, you can use a specific chamber, coupled with fluid dynamics to capture cells one by one. Once one cell is captured, you let the other pass through it. This is what has been used quite often, and quite a few companies has been spin off using more or less similar ideas by doing so. Second is you follow Poisson statistics. You setting cells into many micro compartments. If the compound size is right about just slightly bigger than the cell, so most well will contain one cell and self-exclusively repel the other ones, right? So that's a way you follow Poisson statistics, but by doing the size constraint, you can beat the Poisson limit, right? So the second way. The third way is just what you mentioned. You can pattern the surface. You can use specific ligand that can be somehow coupled with cell surface receptor to actually adhere cell on top of it. So people have been using this. For example, Church Group, probably in early 2000, late 90s, 1990s, they've been actually published quite a few papers doing this cell surface patterning and cell culture. So yes, absolutely possible. Yeah.
Okay. Thank you very much. Thank you.
Okay, Haotian, I have to move on, and you save your questions later. Yeah. So we have water. So water. Yeah. Now you are in West China Hospital of Sichuan University. I think that's one place where famous, for the medicine development and all these things. Can you tell us something about the new affiliations and any comments to Yanyi? So please, Walter.
All right, everyone. Thank you very much, Alex, for inviting me to attend this very exciting forum. Particularly, I enjoyed every moment Professor Huang's talk. A very exciting talk. Congratulations! I really enjoyed it. Yeah, I just come back to West China Hospital of Sichuan University because my background is more semiconductor, biosensor, and engineering background. I'm actually quite excited to be able to work closely with doctors in the hospital, try to solve some real problems, hopefully. So my question for Professor Huang, I'm particularly interested for the part you work, about the digital PCR by using the looped amplification method, right? Yeah. My question is, we know that LAMP is not as efficient in terms of amplification. Many applications require 10 copies in the system. But for digital PCR, you basically every droplets you only have one copy, right? So
yes.
how do you address that? Seems like the NAMP is not that sensitive for single copy application.
Yeah, it's a very, very important question, I have to say. Yes, LAMP, conventional LAMP, is actually not that sensitive compared with qPCR. qPCR probably can amplify a few copies in a single tube, but LAMP requires a few more copies. However, when you reduce the volume of a reaction from the microliter to nanoliter to picoliters, then the effective concentration of your reactant has been increased by thousands to millions fold. So under that conditions, you actually get chance to amplify single copy reactant. Of course, you need to slightly tweak the reaction conditions. For example, you probably need to tweak the clotting reagent a little bit. You probably need to increase the concentration of the primers higher, like twice or 3 times higher. So that small tweaks wasn't that fundamentally important, but it helps. But fundamentally important thing is you actually reduce the reaction volume for every single copy. So it gives you a chance to force them to react. So in digital LAMP, the reaction time doesn't extend it. You can still just wait for like 2,000 minutes, then get all this done. So yes, it can work for single copy.
Okay, got it. Very exciting. My second question is about your RNA sequencing. The way you created for the library preparation for RNA. My question is, does this type of preparation method, is that compatible with a major platform like Nanopore or Illumina or BGI type of machine? Or it's kind of more favorite for one type of platform?
I would say yes and no. So single Nanopore doesn't require this kind of sample prep, so we never thought about using this TM5 transposase to prepare the library for Nanopore. It doesn't require that step, so it's completely different track. So no, but Illumina and BGI machine, yes. However, the BGI sample prep protocol does require very specific adapters, and also their application methods is different. It's more or less like a hyperbranched MDA. So it's slightly different. If we have to use this RNA sequencing protocol, we need to actually do a library conversion for them to be used for BGI. So for Illumina platform, this method, Sherry or Minerva, can be directly adapted to Illumina sequences without any further changes. The adapters will be identical. So if you want to do it as fast as possible, enjoy the 4-hour sample prep, then you use Illumina at this moment.
Okay, yeah. So thank you, Walter. Save a little bit of time. Yeah. So Xinyu, before you ask a question, I ask the questions from the audience. So several audience leave the questions here. How to prevent the test target organic material from pollute the silicon? In other words, is the silicon disposable or can be used multiple times?
Oh, I think they're trying to ask in this mica, like small gas plate. It's essentially it's a silicon dioxide, right? So can you get polluted from the silicon? No, we don't see that. It's a highly pure silicon dioxide we use for material. It's like you put in the fiber, optical fiber, so it's highly pure material. It doesn't really cause the pollution for PCR. However, we were worrying about if this silicon dioxide will absorb certain nucleic acid and create the false negative. So luckily, with the surface treatment of the hydrophobic staining using CVD, then apparently the DNAs wouldn't stick on this hydrophobic surface, so we don't see the molecule loss in our system while we're testing that. So no pollution and no sticky on the surface. No.
Okay, very good. Yeah. So Xinyu, now it's your turn.
Sure. Thanks, Alice. Thanks, Yanyi, for the great talk. I think it's very insightful to show us the whole path you have pursued from very simple design to a very complex design for this digital PCR platform. So my question is regarding to that. Sorry, I missed your first from 5 to 10 minutes because I got disconnected. So my question was, I don't know if you talk about anything, is how do you deal with the sample prep, sample preparation of the single cell sequencing using this digital platform? Is there any specific steps you have to pay attention to? Because we talk about contamination free, but definitely the sample prep could be one of the major steps you want to pay a lot of attention, right?
Yes, you're absolutely right. The sample prep is a very crucial step for single cell study, no matter it's the sequencing or it's just the nucleic acid quantification. I'll give an example how we typically do this. We've done this by starting by picking single cells or preparing single cells. Two major approaches we used has been proved very useful. One is it's not many single cells, say 20, 50 single cells. Then you do manual pipetting or mouse pipetting. You pick one by one under a microscope. Then after you pick it, you actually need to redistribute this cell suspension into a clean buffer, wash it, and pick up again, and wash it, another droplet, and another droplet, and another droplet, 3 or 4 times. Then all these contaminants outside of this cell, sticking on the surface or in this original suspension, has been diluted and cleaned out. So that's one thing. Second, if you are dealing with thousands of the single cells, or at least hundreds of single cells, then apparently like labor-intensive work doesn't work that well in this scenario. So we actually use flow cytometer to actually dispense one cell in one well fashion into 96 well plates. So by doing so, you actually, first you prepare receiving plates by adding the lysate buffer into every well of the 96 well plate. And then you just directly flow cytometer to shoot the single cell into the plate. After they reach the plate, they lies there. So that's the way we do the single cell preparation. And after that, things became much easier. All you worry about is you don't want RNA to be staying at a room temperature or at ambient condition, even 4 degrees for a long time. So you need to make sure to convert RNA into DNA or into a library as quick as possible. Then this DNA product can be stored there for a long time. It wouldn't really change that much. So that's the trick. So cleaning, preparing the sample in the right condition, and then prevent RNA degradation. There are 3 major considerations for most of the experiments.
Thanks, Yanyi. Maybe just one last quick question was, have you thought about like making an integrated either a device or a chip to kind of do all this series of sample preparation steps like automatically? Any thought on that or any efforts on that?
Yes, it's a very good question. We thought about that, but unfortunately, we haven't really done any complete project like this. We thought about there are two major schemes you can work on this automated sample prep. We like to call it from sample to sequence or from sample to result, right? So one way is to utilize this advanced robotic existing robotic expertise, right? So liquid handling and transferring has been developed for decades. You can collaborate, collect all these tools and put them into a pipeline. Then you sling plate one by one steps, go there to do so. So if you're running a big sequencing research center or platform, that's probably the easiest way to do so. You can purchase commercially available tools or machines to dealing with from picoliter to microliter liquid handling, dispensing and transferring, and couple them together. It will be fine. You just need to couple your protocol into this machine. Totally fine. Second approach will be, I personally would like it more, but will be way more challenging, is to try to put everything on a small, at least handheld sized device. We typically call it cartridge in many applications, right? So can you build a cartridge to taking sample and spitting out the say library at least, then you can go to sequencer directly, or better give you the sequence. We think scientifically totally doable, right? But engineering-wise, we haven't got any progress in my own group because it's kind of too engineering. So sometimes it requires a lot of expertise. So you typically junior students, they don't have such expertise to work in there for a long time. So in my group, I typically don't work on this type of engineering work. I'll leave this to some students. They probably have this internal anxiety to do entrepreneurship work, as they probably will go to do this. Yeah.
All right. Thank you, Yanyi. Great talk.
Yeah, that's very important. So you need to have some students go to the industry or do commercialization, right? Yeah, you need more hard work on that. So here is one question I think is for all of us. Say that now this topic is one of the hot topic, and this area is a crazy area for maths. Yeah, for the latest MEMS research. I think now, for almost 5 of us were based on MEMS. Now, yeah, most of us was changed or move on to this microfluidic or biomedical field. So, yeah, can you comment on this? Is this was a future trend for MEMS? We will all follow this? Okay, Xinyu. Yeah, you are the most youngest one. You first.
Thanks, Alice. I think this is a great question, probably from a student. I think maybe I talk about my experience. I think initially I did do a lot of like a microfab and micro device design, and also a lot of silicon device design and fabrication. But later on, I moved a little bit to like microfluidic devices because of the reason that I want to work on the biomedical field and use my technology to solve those pressing problems in biology and medicine. I think that's the major reason. I think the tool we use is just the hook to the tool. I think I care more about the questions, but definitely going back to the technology, we want to design new technologies to enable new studies in those fields. But I think it's not really like we kind of navigate, we escape or navigate away from our initial area, but also I believe microfluidics and the MEMS are interconnected a lot, right? So in terms of fabrication, device design, and even MEMS, we also have bio-MEMS, like people use those solid state devices to target biological and medical questions. I think from that perspective, maybe the student or the audience who asked this question, I just want to share with you about my opinion is on some day, if I found there is a problem we can solve using hardcore silicon MEMS, probably I'll come back. I think those are all connected. This is also kind of this interdisciplinary research. I think that's a little bit of my 2 cents on this.
Okay, thank you very much. Still very encouragement for all these traditional MEMS, person, folks, listen to that. Also, Walter, how about you? Yeah, I saw you are training in EE, right? You are working more than 20 years in the semiconductor and the related microstructure, nanostructures. You also build up your company sometime, right? And now can you give some advice on this for MEMS trend going on?
Sure, I think my answer to the question is positive because from my experience, I think there's quite a wide opportunity for engineers and scientists in MEMS or semiconductor device or processing can find exciting opportunities in application in medical. For example, semiconductors have a lot of interesting functional material that can be made very sensitive and reliable sensors. And the biosensing is a fundamental technology for medical diagnostics. It's a driven technology. I think that's one part. Second, and we see that, like Professor Huang's talk, the precision manufacturing technique on the wafers or glass plastics can be very useful because the mediator of the reagent system and mediator and integration with the hybrid material and the systems becomes really more and more important in the future medical devices. I think from my own experience, and particularly in medical diagnostics, particularly for molecular diagnostics, the detecting of protein, DNA, and RNA, and look at their, particularly to the single molecule level, and will require a lot of this precision manufacturing device and this type of experience that can be, we can naturally apply what we learn from engineering, from semiconductor to this type of challenge. And the future, I see that it's becoming more and more merged interdisciplinary area from engineering and medical. That's why in West China Hospital of Sichuan University, our president and director of the hospital will always encourage us. We call it Med Plus, medical plus engineering is a major strategical research direction of the school. So yeah, that's my own experience. I'm just excited to see.
Very great. Actually, there was one greeting message to you from your previous students, as Tao Li was in the Southeast University, is in the audience. He sent a message to me. How nice to see Walter Wu on the stage. He said, his students, in UT Dallas. So say hello. And also he wants to know more about your new position and new center. Okay, yeah.
Oh, okay. Yeah, thank you. Yeah, hi, Tao Li. Great to meet you here virtually. So yeah, my new position is in the Precision Medical Center. I'm the associate director for the center, and also in charge of the new Center for Future Medical in terms of biosensing and diagnostics. So my role is covering just similar to what Professor Huang is doing. We are pushing medical diagnostics molecularly to a single molecule level, like single molecule DNA, that's digital PCR, then single molecule protein, and for very high sensitivity detection of proteins, and also the single cell, right? And this is some my future research going to be pushed to single particle type of medical diagnostics.
Okay, great, Walter. Yeah, so welcome back. Yeah, so yeah, now Yanyi, I give these questions to you as actually you may be, the first group for microfluidics to do all these issues. Now you are leading this trend, and your background is quite, wide because you're training in chemistry, and you go to physics school in Caltech for postdoc, and you do microfluidics. So how can we do that? We didn't train in all this kind of wide background. We are not so smart. Yeah. So can you give some comments to the, the people like me, one single background or someone?
It's not because I'm smart, it's just because I'm dumber, so I couldn't really find a job. I need to do more and more post training, so until I can find a job. And also, you helped me to find a job, so thank you. You are the one who actually triggered me to
I'm a baller.
So it wasn't my intention to train in all these fields. I was actually quite confused and didn't really know what to do at the time. So I explored a little bit to different fields and figure out what I can do and what I really want to do finally. So I didn't learn any biology systematically until I realized I had to do that with my own students. So I became professor. I cannot really black out, so you just need to do it, right? So for microfluidics, I think it's a concept. It's not a terminology to define what kind of technology can only be used or can be applied to microfluidics. It doesn't have to be MEMS. It doesn't have to be analytical chemistry. It's a concept you want to deal with a small volume of liquid. So people typically will think about lab on a chip using the conventional MEMS technology to do microfluidics. I did the same for years, and I'm still doing some of this now. And finally, you realize that sometimes you probably need to think out of the box. You don't really need to put everything just on a chip. All the philosophy and also the physics never changed, right? How you control small volume liquid, it never changed. You still use those kind of chemistry, physics, and also answer certain kind of the biology questions. But the format can be changed. So whenever you've done before, it probably give you some experience you will think about later. For example, when I do the mica, I never realized that the time I was at Caltech doing photonic crystal fibers, those training background actually gave me the idea how to pull the fiber, the glass rod into the mica plates. So you never know. I was open to any technology and ideas and embraced all them. So I think probably that's because I just slacking a little bit. I didn't really try to be the fastest one to chase to certain technology. So I can sit there, watch everybody playing with all kinds of technology and figure out probably something I can use for different things. So I just sometimes be lazy.
No, you're always studying, studying new things. Okay, Hao Tian, you are asked challenges. Now I give you the last chance to ask the questions. I know you are really nervous about your career or all these things.
Yeah, I feel still. I want to thank Professor Huang for the wonderful talk and all the answers to my question, and also your comments to the previous questions are also very inspiring. And here, maybe I'll ask a final question about what, like in the future, what aspect you think is like most promising, like a topic, like where's the people like me, like with the microfabrication background, is very worthy to explore. So maybe it's a very greedy question, but yeah.
No, here's actually it's a big question. I couldn't really say I ask myself every day, but I ask myself quite often what to do before I retire. So in the next, say, 10, 15 years, what will be my main topics, or what will be the main topics I want to inspire my students? I cannot really say for sure what particular one project, but I really like is try to bridge the information world with the fabrication, with biological system. So it sounds very vague, but it's true. I try to realize, is it possible you can actually process your conscious within an artificial system? Can you actually build a system can mimic your conscious formation? Like your brain function can be vividly resembled in a device. Is that possible? It doesn't need to be microfluidic. It probably has some certain function, microfluidic, to serve this purpose. But is it possible? I don't know how to do this. So this will be the open question. So in the most time, I was still working with the students working on measurement. I believe, I completely believe, like the accurate and precise measurement, it's the key factor to make the real contribution to all disciplines of science, no matter it's physics, chemistry, or biology. So you have to push the limit of the measurement to continuously moving the frontier of the measurement. Then you can realize there's something new there and allow you to explore more. So that's why, although I cannot really find a good, simple solution for the big question I'm trying to answer, I'm really excited about, but in daily operation, I'm still pushing the limit of the measurement. That will be the way probably can help me to find the solution.
Oh, thank you very much. Thank you very much.
Thank you for asking.
Okay.
Yeah.
Really great questions and great answers. I think we from the Mother Nature, and we try to figure out how beautiful it is, and we try to figure out, how to feel it exactly, right? Try to, mimic it exactly, and this will be a long-term, project. So everyone working on this, and everyone should put efforts on this. Thank you very much. Yeah, time flies. It's almost to the end. Now, thank you all of you, and get on the panel. And I think we really have a very great discussion today. And thank you all of you. Yeah, Yanyi, this is the certification to you. Actually, I hope I see you in person. Yes, sometime. I don't know. In the campus, I will deliver this to you. Yeah.
We will see each other. We are actually working in the same university. It shouldn't be that hard.
Yeah. Oh, but your time is really, occupied. Yeah. So, okay, I will see you. I will deliver this to you in person. Thank you very much for your wonderful technology.
Thank you for having me. Thank you all.
Yeah. Yeah. Okay, bye. Yeah. So this is last week in June. So we almost finished these 4 speakers. And who will get on the stage in July? We have 5 speakers in July. So yeah, all of them from different part of the world, and wonderful talks, wonderful speech. So welcome to stay with us, and I can ask talks. And next week we will see Professor Antonio Facchetti. He's from Northwestern University in US. He's also try to transfer his technology to a company. Yeah, he will share his story, in flexible electronics, organic electronics. So everyone, welcome next Friday. We will see all of you next week. And thank you very much. This is enough today. Oh, bye-bye.